Cooked versus raw rice in soil. One leads to a microbe explosion in 48 hours. A handful of rice can reveal something most gardeners never see. Within just 48 hours, soil microbes can explode in number when the right food source is added. But here's the surprising question many gardeners never test. Does raw rice wake up those microbes faster, or does cooked rice trigger a stronger microbial bloom? The answer might completely change how you feed the invisible workforce living in your soil. If you enjoy practical experiments and real gardening secrets like this, make sure you subscribe to Grow Bountiful so you never miss the techniques that turn ordinary gardens into thriving ecosystems. Understanding how microbes respond to simple organic inputs is one of the most powerful tools a gardener can learn. Soil life is the engine that drives nutrient cycling, root health, and plant growth. When microbes are active, nutrients locked in the soil become available to plants. When microbes are dormant, even fertile soil can behave like lifeless dirt. Rice provides a simple way to observe this activity because it contains starches that microbes love to consume. Why soil microbes respond to rice? Soil is home to billions of organisms. Bacteria, fungi, protozoa, and beneficial microbes all work together to break down organic matter. Their primary fuel is carbon, and starch-rich foods are an easy energy source. Rice is almost pure starch. When introduced into soil, microbes immediately begin breaking it down into simple sugars. These sugars fuel rapid microbial growth, which is why rice is often used in natural farming techniques to capture and cultivate beneficial microbes. However, the form of the rice matters. Raw rice and cooked rice behave very differently once they enter the soil environment. What happens when raw rice enters the soil? Raw rice grains are dense and tightly packed with starch. The structure of the grain is hard and dry, which slows down microbial access to the nutrients inside. When raw rice is placed in soil, microbes must first soften and break down the outer layers. Soil moisture slowly penetrates the grain, allowing microbes to begin digesting the starch within. This process takes time. In most soils, noticeable microbial activity around raw rice begins after about 48 to 72 hours. Fungi and certain bacteria are usually the first to colonize the grains, because they are better equipped to break down tougher organic structures. Raw rice therefore acts as a slow-release microbial food source. Instead of triggering a sudden burst of activity, it feeds soil life gradually over several days. This slower breakdown can actually be beneficial in some garden situations because it provides sustained microbial stimulation rather than a short spike. So what happens when cooked rice enters the soil? Well, cooked rice tells a completely different story. Cooking breaks down the structure of the rice grain through heat and water. The starches inside become gelatinized, meaning they absorb water and soften. This process makes the starch immediately accessible to microbes. When cooked rice touches moist soil, bacteria quickly begin consuming the exposed carbohydrates. In many cases, visible microbial activity appears within just 24 to 48 hours. You may notice a thin white fuzz forming around the rice. This is usually beneficial fungal growth, indicating that microbes are actively digesting the starch. Cooked rice therefore acts as a rapid microbial stimulant. Instead of waiting days for microbes to penetrate the grain, the nutrients are already unlocked and ready for immediate consumption. For gardeners trying to quickly activate soil biology, cooked rice often produces faster results. A simple rice soil test is something gardeners can easily try. One of the easiest ways to observe microbial activity is by performing a simple rice soil test. Start with 2 small containers of healthy garden soil. Each container should hold about 2 cups of soil. The soil should be slightly moist but not soaked. In the first container, bury 1 tablespoon of raw rice about 1 inch deep. In the second container, bury 1 tablespoon of plain cooked rice at the same depth. Cover the containers loosely so air can still enter. Keep them at room temperature and avoid direct sunlight. After 48 hours, gently dig up the rice. In many cases, the cooked rice will already show visible microbial colonization. You may see white fungal threads or a soft breakdown of the grains. The raw rice usually remains mostly intact at this stage, with only minor microbial activity starting to appear. This simple comparison reveals how quickly microbes respond when food sources are readily available. Using cooked rice to stimulate soil life is honestly a handy trick. Cooked rice can be used intentionally to boost microbial activity in garden soil, especially in beds that have been depleted or recently disturbed. The key is using small amounts so the soil ecosystem stays balanced. A simple microbial booster can be made by mixing 1 cup of plain cooked rice into 10 liters of compost or mature garden soil. This mixture can then be lightly scattered across planting beds. Apply approximately 1 cup of the mixture per square meter of soil. Lightly rake it into the top inch of soil and water gently. Within a couple of days, microbial populations begin increasing as they feed on the starch. This approach works best when soil already contains organic matter and healthy microbial communities ready to multiply. Why moderation matters, you might ask. While rice can stimulate soil life, too much can cause problems. Excess starch can lead to rapid bacterial growth that temporarily depletes oxygen in the soil. This can create unpleasant odors or encourage undesirable microbes. Keeping applications small ensures that beneficial organisms dominate the process. Using 1 cup of rice-amended compost per square meter is a safe amount that feeds microbes without overwhelming the soil ecosystem. Now let's talk about the role of fungi in breaking down rice. Fungi play a critical role in digesting complex carbohydrates. In many soils, the white growth that appears on rice is fungal mycelium spreading through the grains. This fungal network is extremely valuable in the garden. Mycelium helps transport nutrients through the soil and forms partnerships with plant roots. When fungi digest rice starch, they convert it into compounds that plants can eventually absorb. Encouraging this fungal activity is one of the hidden benefits of using rice as a microbial stimulant. When raw rice is the better option. Even though cooked rice works faster, raw rice still has advantages. Because it breaks down slowly, it provides a longer feeding period for microbes. This slow release effect can be useful in compost piles or mulch layers where gradual decomposition is desirable. Raw rice can also be used in microbial capture methods where gardeners encourage beneficial organisms to colonize grains over several days before transferring them to compost or soil. In these situations, the slower breakdown actually helps maintain microbial diversity. The real takeaway from the cooked versus raw rice experiment goes far beyond rice itself. It demonstrates how soil biology responds to available energy. When microbes receive accessible carbon sources, their populations expand rapidly. When food is harder to access, growth slows down. This principle applies to many organic gardening inputs, from compost and mulch to plant residues and natural fertilizers. The more gardeners understand these relationships, the easier it becomes to guide soil ecosystems toward greater fertility and resilience. Healthy soil is not built only with nutrients, it is built by feeding the living organisms that transform those nutrients into plant food. And sometimes something as simple as a spoonful of rice can reveal just how alive your soil truly is. If you found this experiment helpful and want more powerful, practical gardening insights like this, make sure you subscribe to Grow Bountiful and share this with fellow gardeners so more people can unlock the hidden power of living soil.